we are speaking about the immune system, particularly the adaptive immune system or the specific immune system. We came across telling that our body, okay, it can defend the microorganism. Here you have to remember, okay, there is what is called as intracellular antigens, okay, that is the microorganism has entered into your cell, and another one is extracellular antigen, that is they are not entered into the cell but they are present in the body fluid. So the fight against this intracellular antigen, okay, this is brought about by the cell mediated immunity. The fight against intracellular antigen is brought about by the cell mediated immunity, okay, that is your T cells. It is brought about by the T cell. Similarly, you will see that if your cells are infected or there is a tumor cells, even these, okay, what happens is your T cell is the one which will come and try to defeat those cells or their infection. But what about those microorganisms or those pathogens or those antigens that are able to multiply in the extracellular spaces? Now see, this is another cell, this is called, what we are telling that it is an intracellular spaces. This is the intracellular, between the two cells, intracellular spaces. Or if your cell itself is infected or if there is a tumor cell, what comes into picture is your cell mediated immunity, and that is the T cell, which we will look in detail in the next section. But what about the extracellular fluid? There also there are pathogen. So the pathogen also multiply in the extracellular spaces of the human body. So this protection is brought about by the B cells, okay, or humoral cell mediated immunity, B cell. So the extracellular infections which happens in extracellular fluids, it is brought about by the B cell. B cell recognizes these antigen. So if you want to know about what are antibodies, we will just go in brief with the B cells. Now you all know that the white blood cells are produced in the bone marrow. So inside the bone marrow, what is present? We have the hematopoietic cell or the stem cells. So now what happens? These stem cells, they undergo changes or modification, or they develop into what are called as B cells and T cells. First what happens? They undergo maturation, which are called as lymphoid progenitor, and from this, okay, we will have what are called as B lymphocytes. We will today just concentrate on the B cells. We have what are called as B lymphocytes, which then produces B cells, and these B cells in turn undergo maturation and develop into the plasma cells and the memory cells, okay. So you see that B lymphocytes are produced, okay. From where they are produced, these B lymphocytes, they are produced from the B cells. These are the progenitor cells. This progenitor cells will form or will produce the B cells. This B cells, what they do? In turn they produce the B lymphocytes. So where is the region of origin? The region of origin is in the bone marrow, wherein they are produced and their maturation occur. Now after these B cells are matured, okay, after these B cells are matured, they get converted themselves into what are called as B effector cells, okay. They get converted into B effector cells. And we have the another cells which are called as the memory cells. So this B effector cell, in turn what it does, it produces the plasma cells, and the plasma cells is what in turn produces the antibodies. This is what we are going to look into today, antibodies structure. So, if you want to know about the antibodies, you should know from where exactly they have come. So, what happens? These antibodies, okay, this entire phenomena belongs to the humoral immunity. Humoral, as I told, refers to fluid, okay, or the liquid portion of your body. So, now what happens is, this plasma cells then produces the antibodies. This you all have already studied in the clonal selection. This antibodies, what they do, they keep circulating in the extracellular fluid, that is in the blood, that is between the lymph, blood and the secondary lymphoid tissue, they will be circulating. Now, what is this antibodies is? These antibodies are naive, okay, that is they are not active. They are active when only when they come in contact with the antigen. So, these antibodies they are naive, okay. Once they come in contact with the antigen, only then they become active. So, in the last class, in the clonal expansion, we saw that when there is an attack from a particular type of antigen, we will be having n number of this B cells, okay. There are many B cells, and what happens, these B cells, they will be carrying what are called as receptors, okay. Just come here, we know that they will be carrying what are called as B cell receptors. There are many types of B cell receptors, and if they find an affinity towards any of the antigen, okay, if they are complementary same to the structure of the antigen, only that particular B cell carrying that receptor will start developing, maturing, okay, and this process is what we told as clonal selection, that is this B cell is being selected to fight against the antigen. So, what is important is, just being a B cell is not an important point, it should be having that particular receptors. So, these are naive cells, okay, the B cells, when they are faced with an antigen challenge, they become activated. And how they are recognize the antigen? They recognize with the presence of the B cells receptors. Once they are activated, they proliferate to the B effector cells and the memory cells. So, then immediately what happens? Antibodies are what? Secretory cells, that is what they are moving around your fluid of your blood or the lymph or the lymphoid tissue. So, these antibodies are very specific to any of or any particular antigen. So, we told that what are antibodies? We defined, like if you go to the previous slide, we can, how we defined it, we told that these antibodies are what? They are immunoglobulin, that is they have a globin portion, that is they are proteins, and since they are produced by the immune cell, they are called as immunoglobulin. The another word for immunoglobulin is your antibody, and what is their function? The function is to recognize the antigen. Okay. What are antigens means? You know, it is a foreign substance. So, this antibody is circulating in your blood or in your body fluid, and it is naive, but once it comes in contact with the correct type of antigen, then they will start to get themselves multiplied, that is they get themselves cloned. And what is important? B cell receptors are important. So, now we shall look into the structure of this antibody. So, when you look at the structure of the antibodies, these antibodies are Y-shaped. Okay. So, what are antibodies? They are glycoproteins. Okay. So, you see that they have this V-shaped, okay, and then a stem, or this is called a stem, or it is also called as a stalk. Where does our antigen, where does our antibody bind to the antigen? We know that it will not bind to the entire antigen, but it will bind to the epitope. And our antibody also, the entire antibody will not go and bind to the epitope, it is only this region, okay, the end of this V region, and this region go and bind to the antigen. So, these are glycoprotein and it has a stalk and it is having a V region, and this is the region which is going to bind to your antigen. So, our antibody will have two antigen binding site. Okay, it is having two antigen binding site. Now, when we look at the structure of this BCR, that is the B cell receptors and the this is the antibody structure. So, they look exactly similar, okay. But what is the difference is, see, if I draw a cell, or we'll get back to this picture, okay. Just think this is your B cell receptor. What is the difference between the antibodies and your B cell receptor is, you will see that these B cell receptors, okay, they are present where? They are present on the hemoglobin, okay, that is the protein structure. What is your antibody? Your antibody are secreted from here, okay, only that is the difference between the B cell receptors and the antibodies. So, once the antigen goes and binds here you can look at this picture, okay. Once your antigen, this is your antigen, once it goes and binds to the antibody, your antibody will become activated. From where your antibody is produced? It is produced from the plasma cell. And what is this plasma cells, from where they are produced? Again they are produced from the B lymphocytes, and they all what? They have the B cell receptor. So, if this receptor to this, if it is bound, your antigen is bound, now the cell will release your antibody so that it can go and bind to that antibody. So, bind to that antigen. Okay, this is the basic antibody structure. This is the Y shape of our antibody, and you know this is the V shape wherein the antigen can bind, and the region where the antigen will bind to our antibody, okay, it is called as the paratope. Okay, this is the region which binds to the antigen. I have written your antigen binding site. So, and this is the stem or the stalk region, okay. So, my antibody is in this shape, Y shape. So, we have 4 polypeptide chain, okay, 1, 2, 3, 4, okay. This is 1, this is 2, this is 3, this is 4. We have 4 polypeptide chain. If you look into the picture, you will see that 2 of the polypeptide chain are long and they are identical, this, and this is called as the heavy chain. Why they are called heavy chain? Because they have a high molecular weight. So, this is the heavy chain. And then we have 2 short chain, this and this, okay. These are 2 short chain, and hence they have a less molecular weight, hence they are called as the light chain, okay. So, we have a heavy chain 2 and 2 light chain. If you look into the picture, the terminal end, okay, since they are polypeptide, what is a peptide? That means they are made up of amino acids. So, what constitutes an amino acid? It should have an amino group and a carboxyl group. Since they are polypeptide, they have a terminal, okay. This is the terminal region which consists of, which is known as N-terminal, that is carrying the amine group. And here you can see at the base, this base, okay, and at this base you can see what is present, carboxy group is present, or the C terminal. So, we have the N terminal and we have the C terminal. And right here, this is your N terminal and this is your C terminal. Each of this light chain, which is the light chain? This one, the red color, okay, that is your light chain. Each of this light chain is connected to H, this is your H, right? This blue color is your H, that is heavy chain. So, each of my light chain is connected to the H chain via a disulfide bond. This is my disulfide bond. Okay. The H chains, okay, this is my H chain. This blue one is my H chain, are connected to each other. They are connected to themselves. How? Via 2 disulfide bond. Okay, they are connected via 2 disulfide bond. Where they are connected? In the mid region. Along with the disulfide bond, they also have the non-covalent bond is also present. So, this region is called as the hinge region. Okay, this region is called as hinge. Very important for your exam. H-I-N-G-E. This is called as the hinge region. So, where this disulfide, 2 disulfide bond of your heavy chain bind, that region is called as a hinge region. Why it is called a hinge region means that is a flexible, that region can be bent, okay. Since it is called hinge region because it is flexible, so that it can arrange into different spatial arrangement to the antigen. That is, we know that the antigen, all the antigens are not similar. There are numerous antigens which are present, and our antibody should go and bind to all this numerous type of antigens. So, what it happens, if it is having a very rigid shape, it cannot go and bind. Hence, you see that the antibody are having a region called as a hinge region which is flexible. This region is flexible, okay. This entire region is flexible, sorry, not here, okay, till here, this disulfide, it is flexible. Only then it can have a definite spatial arrangement and it can go and bind to your antigen. That is, it is able to rotate, it can bend. And this, okay, this stalk region, it is called as the FC region. This okay, this part is called as an FC region. This region is called as the FC region. Why it is called as FC region and why it is called as this part, this above part is called as the FAB. See, I mentioned FAB here means this entire till here is the FAB, okay, and this is your FC. Why it is called FAB and why it is called FC, I will explain in the next class. For time being, just remember this. So, to each of this, okay, to each of this heavy chain is here, it is represented in the blue color, there is a short carbohydrate. See here, I have written carbohydrate here, there is a carbohydrate being present. Now, what is the function of this carbohydrate? Why it should be present? Now, what is the function means? It increases the solubility of the immunoglobulin, so that you know that the immunoglobulins are present in the extracellular fluid, and our antigens also, some of them are soluble antigen and some of them are particulate antigens. So, this carbohydrate enhances the solubility of this entire immunoglobulin. Now, we will look only into the light chain. This is the red color is my light chain. So, the red color and the blue color, which is the heavy chain, at the terminal end what they are having? They are having amino group, that is, they are called as N-terminal. Now, just look only at the red color light chain. These light chain, they have a molecular weight of 25 kilodalton. Okay, they have a molecular weight of 25 kilodalton. And they have approximately 220 amino acids. Just don't go now along with this 110, I will tell you later with that. The light chain, okay, this and this, they together they weigh 25 kilodaltons, and they are having how many amino acids? Both together they have 220 amino acids are present. Why amino acids? Because they are glycoprotein. Why glycoprotein? Because it is a protein molecule. Antibody is a protein, and why glyco? Because here the carbohydrate is sitting, hence it is called as glycoprotein. So, the light chain have a molecular weight of 25 kilodalton, and they have together they have a 220 amino acid. So, this light chain, okay, this is another picture I have taken. See, this is for example, here in this picture, just it is, you can see this is the heavy chain. Whenever it is longer, that is the heavy chain. Okay, here we have put this is as a light chain, just a little bit of variation. So this light chain is of two types. What are the types? There are kappa chain and the lambda chain. Light chain is of two types, whether this light chain or this light chain together, they are 220 amino acid and there are two types, kappa and lambda. These are the two types of light chains. So these two are similar in structure and function, but they are coded by different amino acid. Okay, they have the same function, they appear, the structure is also same. Then what is the difference? The difference is that kappa and lambda are coded by different chromosomes or different amino acids they have. That is, you see this kappa chain, it is coded by the chromosome number 2. Okay, this is chromosome number 2 is what codes the kappa chain, and lambda chain, it is coded by the chromosome number 22. It's simple to remember, 2 and 22. So we have this two chains. Even humans know, like we will be having both kappa and lambda chain, but what is the percentage of this kappa and lambda which we have more means we are having 60%, okay, of our immunoglobulin is made up of kappa chain and only 40% is made up of lambda. This is 40% is present in our body, okay, and this kappa coded by chromosome 2 we have 60%. That's the difference. Now we shall look into the heavy chain. Heavy chain means what the picture should come into your mind, exactly the Y shape. Okay, this, see, this is your heavy chain. Light chain means just remember it is short. Light means it is very light, so it is having less molecular weight and it is just a V shape. See here also the same thing. Heavy chain we have drawn it here inside. Okay, this gives you the exact Y shape, and you see here light chain just a V shape. Now we will come to the heavy chain. This heavy chain, okay, it has a molecular weight of 50 kilodaltons. That is this part, okay, this entire part is 50 to 70 kilodaltons. 50 to 70 kilodalton. Okay. Whereas a heavy chain, light chain, how much it was? Only it was 25 kilodalton, but a heavy chain is 50 to 70 kilodalton. And here what happens is, now we told light chain is of two types, kappa and lambda. They are similar in structure and function, only their the chromosome which code them is different. Okay, one is coded by chromosome 2, the other is coded by 22. Now what is the different type of heavy chain? The different types of heavy chains, there are 5 different types of heavy chain. Okay, see, the heavy chain consists of a constant region, we told. Okay, so there are 5 different types of heavy chain, and how they are written is, they are written in Greek letter. For example, this is gamma, this is alpha, we have mu, okay, we have delta, and we have epsilon. These are the 5 types or 5 classes of the heavy chain, and they are represented in the form of Greek letter gamma, alpha, mu, delta. In English I will write it as G, A, mu is M, delta is D, okay, epsilon is E. Okay, how you can remember? See, GAME is game, but what has happened? There is change in center, D is sitting, a game change. Okay, so you can remember this. Next we will go through other parts of our chain. Now our light chain and our heavy chain, okay, this light chain and the heavy chain have 2 distinguished region. And what is the distinguished region? That is, they have what are called as variable region. You can see here, we have written, this is the variable region of VL means variable region of light chain, and this is variable region of heavy chain. So, here also we have the same thing. We have the variable region of the light chain and variable region of the heavy chain. So, this variable region, look here girls, here also we have written VL and VH. This variable region, okay, they have how many amino acids? They are having 110 amino acids. Both this variable region of heavy and light chain together they have 110 amino acids, and they also have an N-terminal region. And then this is the region which is very important, because this is where your antigen will bind, and hence it is called as the hypervariable region. For example, the light chain is called as a light chain hypervariable region, and the heavy chain is called as a heavy chain hypervariable region. This is where exactly your epitope will go and sit. This hypervariable region, they have another term also. It is called as complementarity determining region. Okay, it is also called as CDR. CDR. Okay. Complementarity determining region, or in abbreviation it is mentioned as CDR. What is CDR? CDR is then another name of the hypervariable region. So, now let me explain it more clearly. You will not have any confusion now. Okay. See, we are talking about the hypervariable region. Okay, this region we are talking. Okay, this region we are talking. So, now you should understand, now if my if this region, this hypervariable region is linear like this, you have three regions of CDR. Okay, so they are represented as CDR1, okay, R1, R2, okay, and R3. Okay, so there are R1, R2 and R3. You can see here, okay, CDR1, CDR2, CDR3. But what did I do? I put it in the linear form. So, only in particular region like this they will be. In the linear form you can see this, it is like this. So, these are called as the hypervariable region, or they are called as CDR. Then what about these region? You can see that green one. No? What are those? They are called as framework residue. Okay, framework, F-R-A-M-E, framework residue. These are restricted variability. Okay, they are not hypervariable. They cannot just go and bind. They are very restricted persons. They are just not going to bind to anything. So here what happens is, this linear, okay, whatever was linear, you can see them, they are bent here. Okay, this region is bent. Now once they are bent, what happens? Whichever these regions, whichever were far away from each other, now when they are bent, they come closer to each other, and then that is why they are called as the hypervariable region of the heavy chain and the light chain, because this is the region carrying 110 amino acid, which is the exact region which goes and binds to the epitope of the antigen. And then, okay, if this is our variable region and we have named it as VL and VH, what are these others? What is this bulges? What you are seeing? These are called as the constant region. Okay, they are not variable. They are just constant. They are not the region which are going to bind to your antigens. Hence, these are called as constant region of light chain and constant region of heavy chain. So, how many constant region are there? Light chain has only one constant region, whereas a heavy chain has three constant region, 1, 2 and 3. Okay. They do not bind to the antigen. It is only this portion, the variable region carrying 110 amino acid with their N terminal of light chain and heavy chain is the one which is going to bind to the antigen. Therefore, the others are called as the constant region.